Okay. So welcome back after this short break. It's my great pleasure to introduce Kavita Jain, who did her PhD in theoretical physics at AFR Mumbai, followed by postdocs, where I think that's when she began to switch and work on evolutionary theory. And today she's going to talk to us about impact of changing population size on neutral genetic diversity. Looking forward to hearing you.
Okay, so good afternoon. So I don't know about you, but I certainly feel very hard to listen to talks and give talks in the afternoon. So thank you. I hope you are better. You're better at this than me. I am. Okay. So I'm Kavita. I am a theoretical physicist by training. I worked in statistical physics, and as Vishu already said, I switched at some point. Not switched, but made some sort of have a coexistence of physics and biology. Okay, and first of all, I also want to thank all the organizers for letting me speak about my work today over here. So I will speak about the impact of changing population size on neutral genetic diversity. So there are lots of words here. I'm sure all of you know some of these words already. You have heard, in the last 1.5 weeks. But however, let me go slow and unpack some of these words one by one. Let's start with the simplest one. Genetic diversity. What is genetic diversity? So let me give you an example. So imagine that I take 4 of you from this room. You have my sample of 4, and let's sequence your genome. Okay, and I know somehow that the ancestral allele we know somehow, and if you have an ancestral allele at a locus or a site in the genome, I put a 0. Otherwise, we put a 1. Okay, so we took 4 of you and did the sequencing, and it turns out that all 4 of you had all the sites had a 0. Right? So at all sites, all those 4 samples were genetically identical, and so clearly there's no genetic diversity in that sample. Clear? Okay, but of course that's not what will happen usually. Usually, typically, because there is mutations that happen, although with a low probability, it's more likely that I'll get a sample of this sort. That means these 4 individuals, they will have some places 0, some sites will have 1, and so on. Right? So therefore, there is clearly, there are some sites where all the individuals in the sample have identical, like for example, site number 1, site number 6, site number 7, but there are other sites at which they are not. So then we will say, of course, there's a non-zero genetic diversity. But that's a qualitative statement, and we all are interested in quantitative statements. We want to know how much is the genetic diversity. There are many measures of genetic diversity. Let me tell you the simplest one. It's a very simple one. It's called mean pairwise diversity. Some of you perhaps know it as a mean Hamming distance. So let me just take you through this exercise. How do we calculate that? So suppose I take the sample again and look at, let's say, site number 2. So what I'm going to do is just do a pairwise comparison. If the allele or the 0, 1, if the 2 individuals are identical at that site, then we put a 0, otherwise we put a 1. So let's just do this exercise. So let's look at the site 2. Compare individual 1 and 0. This is 1 and 2, this is 1 and 0, so I put a 1 here. Compare 1 and 3, they're identical, so I put a 0 here. 1 and 0, I put a 1, because they were different, and so on. So there's this 6 pairs, 1, 2, 1, 3, 1, 4, 2, 3, 2, 4, and 3, 4, and I just put accordingly. If they were identical alleles, I put a 0, otherwise I put a 1, because they were different, right? So that's my diversity at the site number 2. I just did pairwise comparison, that's it. So this is only at the site number 2, and I just repeated, assuming things are homogeneous and so on. I just average it over all the sites. So this is my diversity, and this is the quantity which people measure. So this is simple enough, right? Okay. So this is the quantity which actually people have measured. And today I'm interested in genetic diversity, specifically neutral genetic diversity. So neutral, all of you know by now, it just means the fitness is same, whether it's a 0 allele or allele 1 is the same equal fitness, right? That's called neutral. So we're looking at the parts of genome that are neutral, and we want to measure this kind of pairwise diversity at those portions or those parts of the genome. So this has been done many, many times, and this is somewhat newish data by this author. And what he is plotting here is the pairwise diversity as a function of the census population size. So he's been able to estimate it through various taxa, and then this is what is plotted here. Okay, the pairwise diversity that I just told you. Okay, so what you find here is that as the population size increases, diversity increases, which is sensible because if there's larger population, more mutations are likely to happen, and therefore you expect more diversity. So that's kind of qualitative thing is all right? So this qualitative trend is fine, but there is a theory. This is the cumulus neutral theory. It's a very old theory, and it says because neutral, of course, there's no selection. It invokes just two parameters: size of the population and mutation, and that result of the cumulus theory is that this pairwise diversity should increase linearly with the size of the population. So which is what is shown over here in this gray band for two mutation rates. So if I just say that you're looking at neutral portions of the genome, we should apply cumulus theory, and therefore the genetic diversity should lie in this band, which is of course not the case. Look at this, the things are different by orders of magnitude. Okay, so our data lies here and cumulus theory lies over here. So this has been an enduring problem for almost 50 years. So this is called Lewontin's paradox. That why is the genetic diversity, specifically neutral genetic diversity, so low? So this has been the question for a long time. Okay, so here is an idea. So the idea is that, look, you're plotting with respect to the census population size, but it's not the census population size that matters. So to give you an example, suppose you have some population and mutations can occur, and a mutation that occurs is a deleterious or highly deleterious mutation. It's almost lethal. It kills the individual. So you started with this population, but many mutations were lethal. So therefore, you have only a small population size which is left, which will give offspring in the next generation. So effectively, your population size is small, right? So the key idea now is that, look, cumulus theory invokes only population size and mutation rate, but there are other evolutionary processes, for example, selection, which are not taken care of here because, even if you're looking at neutral genetic diversity, the whole genome is interacting with other things, selection, migration, and so on. So we have not taken care of that. But let's assume the effect of all those evolutionary forces into this population size called the effective population size. So for example, here when I said lethal mutations, effectively the population size is small. So in general, all the other complicated processes that are happening, let's put somehow all the effect into this effective population size. Okay. So that has been the basic idea, and biologists are very enthusiastic about this effective population size concept. So because then you just have two parameters, the mutation rate and the effective population size, and then you can just apply cumulus theory. You're back into that same old theory, right? So that's great. In fact, they measure effective population size. So how do we measure it? Simple. So the pairwise diversity is equal to the neutral mutation rate times the effective population size. That's a simple formula that comes out. Pairwise diversity we can measure, as I explained in the first slide. Mutation rate you have to measure experimentally, and of course then you can find the effective population size by dividing the two. Right? So people have done this, and for humans, for example, the population size that matters is not 10 billion, it's just 10 to the 4. Whereas for the Drosophila, it's like 10 to the 6 and so on. Right? So effectively what matters, according to this theory, is a smaller number. And therefore we are basically on the, more on the left-hand side of this axis. Okay, so that sounds good. Yes, question?
Can you explain why, I don't know what population are you looking at for Drosophila and for the humans, because I would expect humans and Drosophila sort of like the effect pop sizes to be similar depending on what the actual population size of Drosophila.
No, it has nothing to do with the actual population. See, the idea is this: you're trying to fit this data point, let's say, over here in this band. That's all you're saying, basically. That's it. And that's what, so, and then you push this point in this band and read off the x-axis. That's the effective population size. Basically, that's it. There's nothing more in it. Okay? Okay. All right. So people have measured this and so on, and they have found all this. And like already, as you asked the question, and the answer was that, basically you're doing a data fitting. That's not an understanding, right? It's just fitting the data and saying that, if I work with this number instead of that number, things will work out. That's not an understanding, and therefore we can reframe the question asking, instead of asking why is the diversity so low, we can ask why is the effective population size so low. So there's been a lot of work for since 50 years, and many mechanisms people have proposed to understand this low effective population size. I'll just tell you the status of this problem is that unfortunately, although these mechanisms help us to reduce NE qualitatively, quantitatively we still don't have a resolution. So we can put all these things, which I will describe one of them. We can put all these processes, yet numbers don't work out, and that's the status of this problem even today. Okay, so this remains an unresolved problem, and if you're interested, you can look up this nice review article by Charles and Jensen. Okay. However, these mechanisms are important and interesting themselves, so we can, keep Lewontin's paradox at the back of mind and look at these some of this mechanism in detail. So I will describe only one of them. So here's the plan. What I want to do is to describe this mechanism called a selective sweep. I will describe what that means, and I will describe to you how the selective sweep affects the neutral genetic diversity in an asexual population. So this work is done with Sachin Kaushik, who was, until some time ago, was a postdoc at University of North Carolina. He's a former PhD student of mine, and he was working with Parul Johari, who's an assistant professor there. Okay, so then I will describe our results and show you how we model things and how and describe our results about neutral diversity. And then I'll ask this question. Okay, there's a lot of interest, and we do a lot of work. I mean, we mean the community thinking, measuring effective population size, but can you always define an E? So I will ask and address some aspects of that question. So first, let me do the slightly simpler thing, which is to first of all define to you this mechanism which takes this selection and other things into account. So one of this mechanism is called selective sweep, and this was proposed just after Lewontin had shown his data and said that, look, why is the diversity so low? So the idea of Maynard, Smith, and Haig is the following. So I will describe it in a very sort of a limiting case. So the idea is that think of just two loci, not the entire genome, just two loci. Okay. First locus is a selected locus, where I have a black, this little a and capital A. And the second locus is a neutral locus. So that's a little b and capital B there. But importantly, these loci are physically linked. They are together. That's asexual. Okay. So let's say initially we have a wild type at the first locus, and at the second locus we have neutral diversity because we have capital B as well as small b. And then at the next generation, let's say there's a beneficial mutation capital A arose here. I know that you've learned here that just because you're beneficial doesn't mean that you're going to spread. You already learned this concept of fixation probability, right? So, but conditioned on fixation, that means it's probability, but conditioned that fixation occurs, what do you expect? You expect that this capital A will spread to the population. That means this population will take over at some point, and everybody will be carried, will have capital A, this green A, at the first locus. But look, what's happening? Because these guys are physically linked, this capital A came with a little b, and when it spread, then all the individuals at the end also carried only little b, right? Because of the fact that it just dragged along. It's called hitchhiking. This took a ride, these little b's, the neutral ones, they took a ride along with this driver mutation, which was the selected one. Okay? So as a result, in this extreme case, you see there is neutral diversity is 0. There's no neutral diversity. In general, there's a lower neutral diversity because of this dragging, because of this hitchhiking. That's called selective sweep. Sweep because it swept away the diversity. Okay, so that's basically the idea of selective sweep. It's one of the mechanisms to understand Lewontin's paradox, to say that why an effective is smaller. And we could generalize this for asexual population, basic idea, but lots of loci and so on, and put in other biologically relevant things, which I'll not describe in detail. But basically, you have this selected locus, A star, it spreads to the population, and at some point you have this. All of them will carry this A star. And when it's asking, what's the genetic diversity at the end of the sweep? In this case, because it's a more complex model, it's not 0, it's some number which we would like to understand. So how do we model this? We want to understand what's happening in the subpopulation. So the key idea here is, we want to, so if you think of this subpopulation, with respect to each other, they're also neutral, because each of them is carrying A star, and all the blue ones are also neutral. So in this subpopulation, everybody is neutral, and therefore the idea is, let's subsume the effect of selection, because of which this population was growing. Let's subsume this effect of this selection through an effective population size, which is changing with time. So you see this population is growing, right? So therefore, we will model this as a population size which is changing with time. Okay, so this is, I can further assume for some reasons that the population is changing deterministically. So I'm interested mainly in understanding stochastic models of evolution. And when we do stochastic models, there is a standard framework of Fokker-Planck equations. In population genetics, it's called diffusion theory. So there is a certain very standard sort of equation that one could write down, which looks like this for neutral population. So there's a second derivative, and inside that is x into 1 minus x. Okay? And how I'm pointing out to you that this coefficient is the population size, which is my effective population size, and this coefficient, however, is time dependent. Okay, and which we are modeling as logistic growth because I'm assuming that n is changing deterministically. So this just helps things to solve, and it can be justified. Okay, so this is the equation, and this it can be used to answer several questions of interest. Right now we're interested in answering the question about diversity. I explained to you a particular kind of measure, which was the pairwise diversity, but there's something more that people measure. It's called site frequency spectrum. Don't worry about it if you don't know it, but it's another measure, okay? So, and it's a bit more, it's much more actually than the pairwise diversity that I explained to you in the beginning. So what we are able to do is to solve this one and get an analytical expression for the site frequency spectrum. And I'll just show you the answers. So this is the site frequency spectrum plotted as a function of the allele frequency, and green curve is our analytical expression, and the red points are from simulations. And you see, it's on a log-log scale. There is a transition in the power law. Initially, it's a power law, and again, it's power law. At short, small frequencies, this power law is it has a power 1, exponent 1, and then it goes to exponent 2. So there's a crossover from this 1 by x, which is a standard Kimura's result, to something which is a faster decaying function. And that faster decaying function has the interpretation of reduced diversity. So I haven't explained to you completely, but that's the interpretation. Okay, so therefore there's a crossover in this site frequency spectrum, and this behavior is universal in the sense that we added more biologically relevant things like dominance and breeding and other stuff, and yet this qualitative behavior, not qualitative, these exponents remained unchanged. Okay, so this was this question about how diversity, individual diversity is changing when there's selective sweep occurs, and basically you can look up other details if you're interested. But for the modeling, we assume an effective population size. Okay, so now I want to answer the second part of the question, which is, can we always define effective population size? I already showed you different data. People have measured it in Drosophila, in humans, and blah, blah, lots of things. Okay, so I'll give you the motivation for this question first of all. So far I assumed that this effective population size was changing deterministically. But in general, and that's because we had assumed that selection is strong, so therefore things will just grow deterministically. But generally, it will not happen. So population size generally will fluctuate. It is also a stochastic variable. So let me give you a specific model that I'm thinking of. So imagine the population size is just changing between two sizes, N1 and N2. So it's N1 sometimes, and then it flips to N2. And then, so this population is expanding, contracting, expanding, contracting. That's all it's doing. It's a demographic. Okay? And the question that we're asking is, can we capture the randomness in this size? So you have two sizes, N1 and N2. Can we capture this randomness by an effective population size? Okay? Let's give you the answer. The point is this. So if you're a physicist, the first thing perhaps will come to your mind is, it's a question of these time scales. You see, if I had a constant population size, I have just one time scale, which is the time scale over which the drift operates, genetic drift operates. And that time scale is of the order of the population size. But now I have another time scale, which is the switching rate, or the inverse of the switching rate, right? So we have a time for genetic drift to act versus time to switch the population size. And it turns out, I'll first give the answer, that if size is switching very slowly, so for a long time, the switch remains, population remains almost constant. Okay, it will maybe in 10 years, it will switch, right? It's basically population size constant problem, right? Okay, but that's one limit. In that case, NE is simply the population size. the higher one or the lower in which we started the process. If it is switching very, very rapidly, every generation or so, it turns out in that case also an effective population size exists, which is given by a harmonic mean. If you know it, great. Otherwise, it doesn't matter. It's just some kind of mean which can still be defined. But if these two time scales are not well separated, they are overlapping, then we cannot define an effective population size. So you can imagine that most of the time, in general, things will be overlapping. Time scales will not be extremely different from each other. They will not be 0 and infinity. There's a lot of parameter space in between, right? Okay, so one has to ask this critically. Can I define a critical effective population size? This question. N1 and N2 can be anything, it doesn't matter, I don't care. So the time for the drift would be of the order of N1 or N2. N2 by N1 is some constant I can fix. Let's say N2 is 10 times N1. So an N1, let's call it N, and N2 is 10 times N. So it's order N, right? And the other one is the switching rate. So those are the two time scales. Yeah? All right. Okay. So that's the answer. But then how do we get this and so on? Actually, these things somehow in PopGen, it's not done much. I mean, it has been done, but not much. But as a physicist, we know these things, at least since the 1990s. Think of stochastic resonance, for example, or many other problems. Okay, but let's think from this point of view. So I'll try to explain to you because, again, because we are physicists, many of you. Okay, so key point here is temporal correlations. So let me try and see if I can explain this. Okay, so the point is this. When you write down the Fokker-Planck equation now, my population size is also fluctuating number. Okay, so it is a whole trajectory of the population size for which I will solve this Fokker-Planck equation, right? Okay, so we can solve this, and we can do a very standard trick, which is of eigenfunction expansion. It can be written like this. But usually we are interested in not just in a single population size trajectories. We want to average over all these trajectories, right? So that's what this angular bracket denotes. But this is a very difficult thing because we have to look at the joint distribution till time t. I'm taking an average of the probability that the population size was n0 at time 0 and n1 at time 1 and n2 at time 2, the whole joint distribution. Okay, this is extremely difficult. I mean, it's almost impossible to do. Okay, but we can do things which I am not showing you. Okay, so it turns out, however, that if you expand this, these are standard things called cumulant expansion. Don't worry if you don't know it, but I will just tell you that there is a way of seeing these things. So one can expand these things and so on. And it turns out, basic point is just this. Because we're looking at joint distributions, if we can ignore the correlations, because today, if I have n individuals in the population, it's not like yesterday it was a totally random number. Most likely it was related to what happened in the previous generations, right? That's called correlation. Okay. If, however, this N1 and N2 is structuring fast. Today is N2. Yes, tomorrow is N1. They're not talking to each other. They're not correlated. Okay. So if I can neglect those correlations, then and only then I can define an effective population size. Okay. So this is a precise mathematical statement. So we, in sort of physically, we just tend to say that when the time scales are well separated, then effective population size can be defined. However, more precise idea is that when the correlations can be neglected, then effective populations can be defined. Otherwise, you cannot do it. Okay, so that's basically I wanted to say, because I haven't shown you much details. I haven't even described to you the full model, but I think that's all right. But I will finish with one thing. Okay, fine. So we cannot define effective population size. All right. But what's the impact of that? What does it do? So I'm not showing here diversity, but I'm showing here the fixation probability as a function of time in this model of N1 and N2. And what's shown here is that suppose population size was changing very slowly. It was fluctuating very slowly. That's a blue curve. Okay, it behaves in some way. All right. And this dashed curve here is when I replace it by a theory of constant population size, but its size is effective population size. So if I do that, then that's the black curve. So they are pretty good agreement. If the fluctuations were very fast, in that case, the red curve, and then there is the effective theory with the dash curve. So they also work very well. And in between is magenta, where I cannot define effective population size. So you see, there's a whole range. These fixation probabilities differ almost by a factor of 2, okay, in between. They're very, very different from these two limiting cases. And it can affect things. Okay, so the answers are substantially different from these slow and fast cases. Okay, so that's, I think, roughly I wanted to say that there is this 50-year-old observation that the neutral genetic diversity is much smaller than when predicted, assuming there is no special structure, there is no selection, no many, many things. And that lets one to sort of fit the data and therefore define the so-called effective population size. But in some situations, at least one should be aware that you may not be able to even define this NE. And in that case, you just have no choice but to develop a full-fledged theory. You cannot simply use cumulus theory by putting a different number. And this is clearly a challenging, challenging problem, because it's not just about NE. People also define effective selection coefficient, effective migration rate, and so on. This is general, problem, and clearly much more work needs to be done. Okay, let's stop here.
Thank you. We have time for questions.
Yeah, we have. That's what this site frequency spectrum that I said, which we can calculate. So certain integrations have to be done, and that gives me pairwise diversity. In certain cases, we can. The conditions under which we can define any pairwise diversity, you can always define. That's not an issue. But to calculate it, I want a simplish model. And the simplish model would be when I don't have to worry about all this complexity due to all these various time scales, migration is happening, selection is happening. Easier if I can just put everything in some effective parameter. But the question is, can I always do that? And I'm pointing out that it's not always possible to do it, especially if the time scales are overlapping of these various processes that keep happening. So that's basically the message.
I have a question. So in many populations, single species, we can approximate the stochastic population as a logistic model with some environmental or demographic stochasticity, right? So you showed a toy example of switching back and forth between N1 and N2. What if we had a stochastic logistic model?
Yeah, so actually there is a model that I'm looking at. So we can do that, but math is much harder. So, but this was a model which is exactly solvable.
But in principle, if we had a stochastic logistic model with a strong noise, maybe the correlations will die off a bit fast, and could that lead to a definition of?
No, I think it will depend upon the parameter regime, right? Because how fast those correlations are decaying relative to population size. So you can have a logistic, stochastic logistic one, right? But then that noise parameter, that rate is to be compared with the rate, which is the inverse rate in the population size. Those two timescales have to be compared. So basically, you have to ask how fast those correlations are decaying. Let's look at the two time correlation functions of whichever stochastic population size you want to consider. Let's look at the two-time correlation function and ask how fast are they decaying. That should give you an idea.
Just this diffusion equation you have.
Yes.
NT. Can the diffusion equation be solved for any kind of NT, or the logistic one is the
this first of all, this is not a usual diffusion equation.
Yeah, of
X into 1 by 6 inside.
Yeah,
sure. Okay. So n of this one is actually solvable for any n of t. Okay. Because this one, this eigenfunctions are hypogeometric functions, okay. Gauss hypogeometric functions, and we know everything. So this is a model which can be solved for any n of t. And that's why I first solve for any n of t and then take an average. Okay. But that average is hard.
So one could also look for the effect of alley effects.
Yeah, that thing so, yeah, sure. Yeah, because this is exactly solvable, this equation is exactly, but you have to worry perhaps with the boundary conditions, but I think that can be, that's not hard.
Any other questions? Okay. If there are no
Okay.
questions, thank you, Kavita.
Thank you very
Let's,
much.
break for coffee now.